We're going to talk about nuclear hormone receptors, just in specific general features and structure and function of them. So first, we're going to have a hormone. This hormone is going to be carried to the target tissue on a serum binding protein, which allows it to diffuse easily across the plasma membrane and bind to its specific receptor protein inside the nucleus. So it's going to travel through the blood on the serum binding protein. It will travel through the first plasma membrane that distinguishes the outside of the cell from the inside of the cell. And then once it's inside, it's going to go ahead and travel through the nuclear envelope enclosure. It's going to meet a nuclear receptor protein right here, which, if it's not dimerized already, it will dimerize once it attaches to proteins. So it will form two of them. And here it's going to go ahead and form an RNA polymerase with a bunch of other different proteins that are not embedded into the membrane. Once this RNA polymerase forms, transcription is going to occur, so the hormone binding changes the conformation of the receptor. It either forms a homo or heterodimers with other hormone receptor complexes and binds to specific regulatory regions called hormone response elements in the DNA adjacent to the specific genes. The receptor attaches co-activator or co-repressor proteins, and with them regulates transcription of the adjacent genes, increasing or decreasing the rate of mRNA formation. So it really depends whether these blue proteins are going to either inhibit or activate the certain gene response that the hormone is going to elicit. So once the transcription factors go ahead and synthesize the mRNA is going to go ahead and go through translation on the ribosome. So this is what will occur. Altered levels of the hormone regulated gene product produce the cellular response to the hormone. So now it's going to form a new protein, and it's going to cause whatever altered cell function that the hormone stimulates or inhibits.